as the RNA which is synthesized is a pre-mature RNA. For maturation of RNA, it has to pass through RNA processing. RNA processing, also known as post-transcriptional processing, or also known as post-transcriptional modifications. Let's see here. As the RNA is synthesized in the nucleus, it has to be released into the cytoplasm for the process of translation. To protect its ends by the degradation by exonucleases, the two processes are performed. One is the capping, this is the capping, and other is the tail addition. In the capping, there is the addition of the 5' 7-methylguanosine. This is the structure which is added to the 5', as the RNA which is synthesized is always 5' to 3'. At the 5', this structure is attached, which consists of sugar, this sugar attached by the phosphate bond, and the guanosine base. And to the guanosine, at the 7th position, the methyl group is attached. So that's why it is known as 7, because at 7 methyl is attached, and guanosine, this is the guanosine structure, and 5' of the messenger RNA, it will be attached. So that's why 7-methyl guanosine will be attached by the enzyme. 7-methyl transferase enzyme will be used for the addition of capping to the 5' and to the 3' poly-A tails. A number of many adenine, as you can see here, poly-A, many adenine nucleotides, a string of adenine nucleotides will be added to the 3' end. So this 5' capping and 3' end, they are attached. This poly-A tail is also used by the recognition by the ribosomes. These are also used for the, when they will release into cytoplasm, they are recognized by the ribosomes. They are also for the recognition by the ribosomes and also to prevent from the degradation by the exonucleases. The other process is splicing. Why the RNA splicing is required? As the RNA is synthesized, it will copy all the sequences, coding and non-coding regions. So the coding regions are known as exons and the non-coding regions are known as introns. So these intervening intron regions are removed by the process known as splicing. RNA splicing will remove these non-coding regions and the remaining coding regions will combine together by phosphodiester linkages. And now this coding region will be now having the code for the synthesis of the proteins. Now this code will be recognized by the ribosomes and will add up the amino acids for the synthesis of the desired and the required proteins. If you see here in this structure, this is now mature RNA which has the cap at the 5' end, that is the 7-methylguanosine, and it has poly-A tails at the 3' end. At the 3' end, poly-A tail is attached. So the messenger RNA processing or the post-transcription modification includes capping, poly-A tailing, and splicing. In splicing, the intervening uncoding regions that were transcribed during transcription are removed, and then the coding regions will again combine together by the process by the phosphodiester linkages. So how this process is catalyzed? This is catalyzed by enzyme known as spliceosome. If you see here, this is the enzyme spliceosome which removes the introns and then combines together for the exons. The intron regions, the uncoded regions are cut out by this enzyme known as the spliceosome. And this process is only specific for the eukaryotes.